So today we will continue talking about basis of immunity and start talking about SARS-CoV-2 vaccines. We'll continue the vaccine thread a little bit in the next lecture, and then we'll start talking about treatments. So last time we discussed briefly innate immunity and T-cell immunity. Just as a reminder, T-cell immunity clears infected cells and can mitigate infection. It does not prevent infection from occurring, generally. B-cell immunity is another arm of the adaptive immune system that serves a complementary role. If you remember, T-cell immunity acts as either in a helper role or by clearing the infected cells. And B-cell immunity acts basically at preventing infection in the first place. So it develops a different type of molecule called antibodies that are developed in response to specific viral antigens, which are typically proteins. And the best kind of antibodies are neutralizing antibodies, which means there are other kinds. So neutralizing antibodies are as it sounds. They bind to viral particles and just flat out stop infection. So for example, they can clog up binding site. The vast majority of vaccines, importantly, target B-cell antibody responses. I'll review the chat. I see there's ongoing chat. We'll, I'll look at it in a few minutes and see if we can catch up, or maybe at the end of the lecture. I just want to get going with this. I apologize for these childish cartoons, but I think they're actually great because they demonstrate that a lot of this can be thought of as building blocks and sort of like Lego bricks. This is from an NIH publication that is cited here, and it's sort of a really accessible introduction to the nature of immunity. So one of the key terms that you will see thrown around is antigen. An antigen is effectively anything that can trigger an adaptive immune response. It could be microbes, proteins, tissues, cells from other people, or malformed cells from your own organism. But fundamentally, it's some molecular feature. Typically, for the purposes of what we're talking about, this is some protein on the surface of a virus. Different viruses will have different things sticking out. Different strains of the same virus might have different things sticking out. And this is an antigen. So you have marker molecules that it gets attached to the antibody. And each antibody effectively is evolved to recognize one type of marker molecule in the surface of an antigen. So an antibody, the useful way to think about it is an antibody is basically a molecule that's evolved to bind to another molecule. And this most typically happens in the protein space. So you have proteins binding other proteins. And the reason I like the Lego analogy is that, you have different slots, different shape slots. There's complicated in 3 dimensionals, but, some antibodies basically don't fit in the slots of antigens, so they will not recognize them. There's a cartoon of everything that's involved in the immune system. Again, this is not an anatomy or physiology class, so I'll just go through it very briefly. So all the immune cells originate in bone marrow. So these are white blood cells. T cells mature in the thymus, which is a special immune privileged site, whose sole function for this to occur. Then you have the lymphatic system, lymph nodes, spleen, various other tissues that transport and process lymphocytes. For example, lymphocytes are exposed to antigens in lymph nodes, right? So it doesn't happen in the blood. There have to be both antigens and lymphocytes have to be in the lymph node in order for the presentation to occur. So here's the picture of a simple lymph node. Sorry, somehow I got, there we go. The chat is stuck in the middle of my screen. It's obscuring the slide. Just a moment. Let's see if I can move it. There we go. Got it. Sorry. Okay. So the lymph node, you can basically think of this as, training grounds for the immune system, because that's where they get, the immune cells get exposed to various antigens and they develop their specificity, multiply, and grow. Again, as a review from last time, T cells is, one type of lymphocytes. They do not recognize free-floating antigens. Their goal is to basically kill infected cells. Their surfaces contain specialized antibody-like receptors called T cell receptors. that see fragments of antigens on the surfaces of infected or cancerous cells. There are many different types of T cells. Helper T cells or TH cells don't actually kill anything directly, but they coordinate immune responses by communicating with other cells. Killer T cells or cytotoxic T lymphocytes directly attack other cells with these granules that have perforins and other enzymes that are basically destroy the target cell. And you also, without going into too much detail, you have to just keep in mind that T cells sort of go through a life cycle. So they start with an immature T cell, which then differentiates depending on the stimuli it receives into different types of T cells. This process of maturation and eventually the formation of memory cells is the basis of long-term immunity. So, you know, a simple, a highly simplified cartoon of what T cells go through is that there's this T cells are mobilized, which is just stated. The mobilization itself is a complicated process that involves a lot of signaling and a lot of staging. That is step one. Step two is they need to encounter a cell that digests an antigen. So there's a whole class of cells that are called antigen presenting cells or APCs. You know, conveniently, it's also an abbreviation for armor personnel carrier in the military. So, you know, my military analogy might be relevant. But yeah, so APCs are antigen presenting cells. So when T cells are they encounter a cell such as a macrophage or a B cell that has digested as an antigen and presented in a way that's recognizable by the T cell, that triggers another cascade. There are lymphokines, which help the T cell mature. All right, so now it's recognized something. It basically says, oh, enemy's here. I got to do something about it. So it's now alerted and activated. It secretes more lymphokines, which basically make copies of the same cell. You know, to basically grow your army and then go on to attack the infected cells in two different ways. So for example, some T cells mature to become killer cells and track down body cells infected by viruses or making other abnormal proteins. And then the other T cells basically recruit more cells like garbage collectors, you know, macrophages and other types of lymphocytes to the infection cell. But the key point again, and we'll reiterate this again next time, because the role of T cell and B cell immunity in response to COVID-19 seem to be both significant. So we need to keep both of them in mind. So, but it always deals with killing infected cells. This is fundamentally different from what B cells do. So B cells, even though they share the same fundamental lineage as T cells, so they're lymphocytes, which is differentiated into different types of lymphocytes. You know, B cells are exactly where T cells, so they're in you know, they spend a lot of their time in lymph nodes. So B cells make and secrete antibodies, right? And each B cell makes one type of antibody, right? So, and it's quite different, because if you think about it, T cells themselves attach to infected cells, so they actually directly are involved in, you know, the killing process. B cell can, you know, once it stimulates, it becomes a plasma cell, then it actually can go into the blood that to produce antibodies. And then the antibodies are molecules that exist on their own, right? They're no longer attached to the cell. So B cells mature into plasma cells that pump out many antibody molecules when stimulated by antigens. So you can basically think of these as ammunition, or, you know, killer, I don't know, killer drone producing cells. You know, they're just factories. They make these antibodies, and antibodies themselves, which are fairly simple molecules, go on and mess up the viral infection. So an entirely different process. T cell attaches to the cell that needs to be killed and kills the cell. B cell doesn't really do much to cells directly. It makes antibodies, which then are able to interfere with pathogen development process. Now, antibodies, and of course, like I said, anything, the one thing that's always true about the immune system is very complicated. So antibodies themselves belong to the immunoglobulin family. So the canonical shape of this molecule, if you ever saw this depicted, would be like this Y, which is a simple antibody. I'll have you I'll show you a blown up picture of this and explain a little bit more of the features of it. But they're different, you know, and they can form more complicated complexes by cross-linking with each other. So these immunoglobulins have different types, isotypes, they're called. So immunoglobulin G, which you might have heard of, this is the most common one. This is the one that's found in the blood. You know, works efficiently to coat microbes, speeding their intake by other cells in the immune system. You have IgM, which is, you know, very effective at killing bacteria. It's sort of early marker. The important thing for SARS-CoV-2 is IgA, which concentrates the body fluids, tears, saliva, secretions in the respiratory tract, and the digestive tract, guarding the entrances to the body. So you can basically think IgA as guards that protect the entrances to your body, and IgG is anything that deals with the inside of the body. And both of those turn out to be important for the consideration of SARS-CoV-2 vaccine design. You have IgE, IgD, so they're actually very, they differ only in a small part of this tail molecule here, but they tend to perform different functions, they have different localizations. But just keep in mind, so IgG, IgM, and IgA are the three important ones. So B cells go through a different maturation and expansion process. So antibodies are triggered when a B cell encounters its matching antigen, right? So remember, B cell makes one antibody to one antigen. You might ask, you know, how does it make its antibody? We'll see that in a few minutes. But let's assume it recognizes, you know, the Lego block that it has binds to the corresponding piece on the antigen. So the B cell will then take in the antigen and digest it. Then it will display antigen fragments bound to its own distinctive MHC molecules. So this is the activation phase. The combination of this of those fragments of MHC molecule will attract the help of a mature matching T cell, right? So you got T cells involved again, which at this point you'll have another signaling cascade triggered. So lymphocytes secreted by the T cell allow the B cell to multiply and mature into antibody-producing plasma cells. This is basically a nascent cell that needs to be triggered, you know, activated, cloned, and then, you know, make a bunch of different plasma cells. So released in the bloodstream, antibodies lock into matching antigens. These antigen-antibody complexes are soon eliminated either by the complement cascade or by the liver and the spleen. Right. So that's where we are. It would be important to notice that there are a lot of sort of concepts that are not directly relevant to SARS-CoV-2, but there are important to understand from the perspective of human health. And one of them is the idea of immune tolerance, which is how does your body make sure that T cells and B cells don't kill things they should not kill. And the reason this is important is when T cells and B cells are developing, a lot of the receptors that they will make are effectively random. So there's there's actually a very complicated process of generation of these receptors. And the reason why you want it to be random and diverse is because you do not know which antigens you're going to encounter. So your immune system needs to be able to respond to things it's never seen before. And one of the ways to do this is to generate a lot of variety of receptors in the hope that one of them will sort of have some recognition of a new antigen. And then there's a separate mechanism that if you have some recognition, there's a whole cascade of processes that is engaged to improve this recognition, which I'm not going to talk about. But the downside of this is if you're randomly generating receptors, there's no guarantee that some of these receptors will not bind to things that your body normally makes. So, you know, for instance, imagine that you accidentally develop, I don't know, an antibody to hemoglobin, right? In which case your immune system will immediately kill all of your red blood cells and you're dead. So that's not a desirable outcome. That means that you could have antibodies or cytolytic or cell-killing T cells that destroy your own tissues. It's actually the basis of you know, various autoimmune diseases. But obviously, this is not something that's happening frequently, or in most people's lives it will never happen in a detectable fashion. The concept of tolerance is concerned with a set of mechanisms which prevent organisms from responding to self-constituents. And this is a long-standing theory going back to a gentleman named Frank Barnett in 1957 called clonal selection. And this also gives you sort of a useful way to think about the immune system. So the assumptions of clonal selection is each lymphocyte has a single receptor with a unique specificity. So a T cell or a B cell is going to only bind one kind of antigen. So that's step one. One lymphocyte, one antigen. Not strictly true, but close enough. So if, in order for the immune response to be triggered, a receptor must be bound to the antigen to activate the cell. So once this happens, it's basically the danger signal. Then the lymphocyte is activated, and then we'll make nearly identical copies of effector cells. And the important part is that lymphocytes that are activated by their own cells are deleted early on. The question is, do the different types of receptors made by the immune system vary among people, or does everyone have very similar receptors? Great question. So T cell receptors so your HLA type, which is something that we talked about last time, is based on the germline, right? So this is something you inherit from your parents, and it's different from different people, typically different between different people. So and the way this, you will most readily recognize this is if you ever need an organ transplant, you will require an HLA matched donor, and most of the time, the best candidate is an identical twin. If you have that, then a sibling, then a relative, and it's only very rare that you'll find somebody that exactly matches you. So B cells are done very differently. So B cells, we do have a little bit of differences in the genes that make up the immunoglobulin loci, but most of the differentiation actually happens after you're born. And what happens is that depending on which antigen you see at what time point, you will have different antibodies. So we could be identical twins and even live in exactly the same environment, but because of sort of randomness and when we see certain things, our B cells are going to be quite different, even though our T cell HLA cells are identical. And there's been really great studies in the last 10 or 15 years where we started, you could actually directly sequence the B cell repertoire. I'll tell you just a tiny bit about it and measure how it is in different people. So here's a simple cartoon of how this cycle goes on. So you have a hematopoietic stem cell undergoes differentiation and genetic rearrangement. So that's step one is basically when it makes its receptor. And this genetic rearrangement is a surprisingly complicated process, which has evolved to create very, very different receptors randomly. So there's a lot of randomness that goes into it. Many immature lymphocytes are produced with many different antigen receptors. So this is step B. There's a separate cascade there that recognizes the lymphocytes that target antigen from the body's own tissues. They're destroyed. If you pass, and this is a very important filter, you don't want to release those guys to where they can do damage. Lymphocytes targeting antigen, and the rest of the cells mature into inactive lymphocytes, so they just sit there and wait for something to trigger them. If something does trigger them, which is a main matching antigen, which is shown here is this external stimulus, lymphocytes are activated and produce many clones of themselves. Sort of this process, which is also not developed in this picture, this is a lineage of B cells from this point on, all these clones, is called a clonotype. And it can actually go through multiple cycles of replication. And there's an opportunity to introduce some variation, called somatic hypermutation. So some of these clones can actually also acquire different mutations to specifically bind to the antigen better. All right. So what is an antibody? So an antibody is a simple molecule that has two complementary chains, the heavy chain, which forms the inside of the Y, and the light chain that binds to the outside of the Y. You can sort of conceptually think about this opening is to where the antigen goes, right? So this is depending on which parts of the sequence are presented here is what it'll bind to. So it's sort of a flexible adapter. This is the constant part, which doesn't vary between antibodies, but this is what determines the isotype. And most of what you basically, if you sequence this, so this is called the Fab fragment, so antibody, and this is the constant fragment. So if you see this abbreviation, that's what it means. And the heavy chain and the light chain are fairly short molecules, so that you can sequence. And if you know the sequence of this molecule, you can sort of, figure out, loosely speaking, what it might recognize. So as far as we don't have a very good sense. I mean, there have been, like I said, in the last 10, 15 years, studies on what different types of B cells are in different people. I'm not going to go into this, but there has basically, a couple of take-home messages. There's a lot of variation between perfectly healthy people, so there's no sort of standard complement of antibodies. You can actually watch disease dynamics. So if you sequence somebody, before, for example, they're infected with influenza, and after they're infected with influenza, you can see that a substantial portion of the immune system is dedicated to influenza-specific antibodies. So what holds the heavy chains together, it's just covalent bonding, I think. I mean, this is just protein-protein interaction. It's, your standard, multi, multi-domain molecules. Anyway, so heavy light chains are fairly short, right? So they're only about 200 to 300 amino acids in length. So they're kind of like a typical gene. They have a constant domain and a variable domain. By definition, the variable domain is variable, and it's composed itself from different building pieces called the V region, the variable region, the J region, the junction region, and the overall genetic structure in which they're put together is variable, junction, constant. The heavy chains also have a diversity region, which is specifically put there to increase the variety of things that they can recognize. So a V region and a J region are molecules in your germline. So on chromosome 6 in your germline genome, there's a large number of V genes, J genes, and D fragments that don't vary too much between people. What varies is how they're put together through this process of rearrangement. So you have, if you go back to, let me just go back to the previous slide here. So this step 1, where it says undergoes differentiation and genetic rearrangement, that's the part where a specific version of this VDJC junction is created, and that determines the specificity of the B cell. And this is done randomly. Not 100% randomly, so some things are, preferentially attached to each other. Just to give you an idea, for example, in mouse, there are 100 known unique V regions and 5 J regions. So if all you do is randomly pick one and randomly pick another, it already gives you 500 possible light chain rearrangements. If this doesn't play, you can go and watch it on your own. I just say, did it? Are you still seeing my presentation? Do I need to switch to a different screen? Yeah, let me the whole point of this is that it's a complicated process that if you've never seen it, you should be, I encourage you to watch it because it shows you sort of the fascinating complexity that we have in our immune system. So here's an example of what these V regions look like. So this is the molecule, and what you can do is you can, each one of those is a unique piece that you could pull together. They have these names called V1-35, means it's family 1, gene 35. And this is the actual sequence of amino acids. And it's going to be annotated. Again, immunologists come up with the most obtuse nomenclature. So, these regions, you can see if you just scan across the sequence, you'll see that some parts of it, like this region here, is more conserved. And some parts of it, like this, is less conserved, and especially at the end. So these, you can take, the way you create something that a B cell will recognize is you pull out one of these genes at random. Then there's also a process that sort of messes things around here. It can insert nucleotides, flip nucleotides. It can basically extend things at the end. And then it tacks on the J region. And then that goes on and becomes a part of the antibody fragment that then recognizes the antigen. And you will see incredible diversity generated in the CDR3 region. So CDR stands for complementarity determining region. And there are 3 of them here. right? So the CDR3 is the one that's sort of the most variable and most responsible for determining what the antibody binds. I'll skip this. So I was about 10 years ago, I worked with a couple of immunologists when deep sequencing was still fairly new and cool, to do one of these direct measurements of what a particular antigen does to the immune repertoire in mice. So this is a little bit of a side story. Although, as far, we don't know of any super antigens in SARS-CoV-2, but there are some other pathogens that do encode these things called super antigens. So let me explain what this is. So if you look at the picture on the right, there are actually 4 different molecules shown. So ignore the red ones for a second and the green one. So just look at the blue and the purple pieces. So this is the fragment of an antibody, right? So this is what it looks like in 3-dimensional space. You have these conserved regions, you have these variable regions, and these purple bits are the variable regions that are designed, that are evolved through this variable mutagenic process to bind a conventional antigen. A conventional antigen will come in from the top of the molecule and bind to this variable region. A couple of things follow from this. So remember, a B cell recognizes a unique antigen. And between 2 random B cells, there's not going to be a whole lot of similarity between what the CDR3 looks like, which means that any conventional antigen will only bind a small fraction of your B cells, or antibodies produced by your B cells. So for instance, if you look at all the B cells that are circulating in your body at the moment, the ones of plasma cells that can produce antibodies, or the mature ones that can produce antibodies, not plasma, and say, how many of them are there because I was infected with the flu, 10 years ago? There's going to be a number of them, but it's going to be a very, very small fraction. So among all of your B cell repertoire, maybe 0.1% will bind to a flu antigen. If they do bind to a flu antigen, then they're going to make lots of copies of themselves and briefly expand in their popularity until they're done with with the infection, but then they'll go back down. Now you have these. So this is what typically happens. But some, especially bacterial antigens, they have this unique defense mechanism where they have a specific proteins whose job it is to mess with your immune system. So they're evolved to bind to a lot of antibodies by ignoring the CDR loop and instead binding to something that is conserved, right? So you have this PPL, this protein L, which binds here to the light chain, and you have in Staph aureus, you have this SPA protein which binds to the constant region in VH, which means that these bacteria can basically immobilize or neutralize about 30 to 40% of your entire immune repertoire, which sounds like, it could be a significant disruption. So with that in mind, so this is, so PPL is this Peptostreptococcus magnus protein L. The reason we know this, it's been characterized in crystallographic complexes with human fragments of antigen binding, and it can bind to a whole lot of different antibodies because it basically ignores their variable part. It binds with highly conserved residues in the framework regions of the V gene. And these contact sites are in the variable region framework sites that have remained unchanged during the evolution of the adaptive immune system, and they're highly conserved between inherited V gene segments. Question is, can people actually develop antibodies? Yes, they can. And can a person get reinfected and would the symptoms be less severe? Yes, that is true. And this is by and large true of the most cases we've seen. I'll show you some data a little later this time. Yes, so there's nothing special about SARS-CoV-2. We develop antibodies to it, and they're effective. And convalescent sera, people that recover, are protected against subsequent infection by and large. Not 100%, but by and large. Remember, I showed you a case where somebody was reinfected with a genetically different virus, but even in that case, it was a very mild infection. So it's a very important point. It's a very good thing. Our immune system does what it needs to do in most cases. But this is what we can actually do. We can, so in this study, that we published in 2012, we actually took mice, we gave them PPL. We had 8 mice, 4 control and 4 that were treated, and just measured what happens to their antibodies. And we knew that antibodies that have this particular gene in them should be removed by PPL. And this is a long story. They do, right? So this was a lot of work for 2 charts. But you're basically measuring what fraction of all the immune repertoire is targetable by this protein L. So in control, it's about 40%. If you treat somebody with mice with PPL, it drops down to 10%. So this is basically, we know what happens, what should happens because we know the structure. Let's see if the sequencing idea works. So it does. This idea of going on and doing deep sequencing on your B cells is called RepSeq, repertoire sequencing. One of the things that we've learned by doing this, for example, older people have lower immunity to pathogens and vaccine responses because their repertoire actually looks different. Sort of, it's not as diverse, and it has other features that sort of indicate that it's not as robust as others. If you have chronic infections like cytomegalovirus or Epstein-Barr, also modulate immune expression and proliferation. Now, in response to the question, do people develop antibodies to SARS-CoV-2? They do. And the process of developing detectable antibodies to a particular antigen is called seroconversion. So sero is blood, conversion means something happened. A viral infection will go through several stages. Early viral infection can be detected by qualitative reverse transcribed PCR viral RNA. So we discussed this, I believe, in lecture 2. And this will continue to, you'll still be able to detect viral RNA probably throughout the course of the infection, maybe some fragments even afterwards. As infection progresses, the immune system begins production of specific antibodies to viral antigens. As this occurs, initially, the level of antibodies is too low to be detectable, but it rises over time. And the very specific point where specific antibodies to a viral antigen become detectable, that's when it's said that seroconversion occurs. So you basically didn't have antibodies prior to seroconversion, you have antibodies post-seroconversion. And this forms the basis for another broad type of tests that people can take for SARS-CoV-2 or other pathogens called antibody testing. And this is usually done either through ELISA or rapid tests. I'll say this now, and I'll repeat it several times. The QRT-PCR, so molecular sequence tests and antibody tests measure two very different things. Qualitative RT-PCR measures whether or not you actually have viral RNA in your system. And this usually is associated with active infection. If you have an infection and you clear it, after some time, there's no more viral RNA. So a QRT-PCR test at some point will be negative. Antibody testing detects past infections. Depending on when it's done, you can have an active infection, while there's still some viral RNA and your body's still working on clearing it, but your antibody will remain after the infection has been cleared. So you can use antibody testing to look for current or past infections. QRT-PCR is much more sensitive. It'll detect infection earlier and with more precision, but it's not going to be able to tell you after, say, 3 months, have you had COVID? The question is, really the process of seroconversion for SARS-CoV-2 is no different from other viruses. It is absolutely no different. There's nothing special about SARS-CoV-2. People would like you to believe that there is, but there isn't. It's just a slightly different coronavirus. I think what's different about it is, how the society handled it. There's definitely no precedent in modern history about that. So the question is, so why is it so hard to develop a vaccine? It's not hard. We'll talk about it later today and next time. It's just, you will see where we are now. And we're actually, as a civilization, we're doing pretty well when it comes to vaccine design in response to what this is. So just, I have no financial interest in LabCorp. There's no reason I picked LabCorp. It's just the first one that I found. Here's an example of how you might find that this is an an antibody test. So this is what you might see as a consumer. So COVID-19, IgG. So remember I was telling you about different types of immunoglobulin. So this is a particular type of immunoglobulin, also known as serology test. Serology, again, is just blood work. Checks for a type of antibody called immunoglobulin G. If you have been exposed to the virus that causes COVID-19, your body typically produces IgG antibodies as part of the immune response to the virus. It can take at least 2 weeks after exposure to develop antibodies. This type of COVID-19 test is for individuals who think they may have had COVID-19 and do not currently have symptoms. So this is basically, have you had an infection in the past? If you've never seen this before, here's a 2-minute description of what a standard test for detecting the presence of antibody is. So it's called an ELISA test. It's an old test. It's developed in 1971. Stands for enzyme-linked immunosorbent assay. And the goal is to basically detect whether or not you have an antibody to a specific antigen. So you start with an unknown amount of antigen, so in this case, SARS-CoV-2. It's attached in a microtiter plate. There are 2 ways it can be attached. You can just, attach it non-specifically, so it just sticks to the surface, or you can pull out specific antigen for the test to perform better. So you have antibodies on the surface that capture the antigen. So this is called the sandwich ELISA. Then you wash in the antibody to see if any of the antibody will recognize this antigen. If it does, as is shown here in the picture on the right, so you have viral sample on the surface. So this is antigen. Antibody with enzyme conjugate attaches to viral antigen, right? So you wash this in, there's an antibody that binds the antigen is going to get stuck on the surface. This purple thing is what you actually detect by light. The detection antibody is linked to an enzyme that can itself be detected by secondary antibody linked to an enzyme. You wash the plate several times to remove any proteins or antibodies that are not non-specifically bound. And the plate is quote unquote developed as a photograph by adding another reagent to produce a visible signal. So that's what it looks like, this blue part. The second picture is the sandwich antigen, where you have an antibody on the surface to which you bind the antigen, and then everything else is like before. There is, so ELISA tests, as you might imagine, this is pretty involved. It's a lab test. I mean, it's not a particularly complicated test, and if you've done any lab, I would imagine a fair number, I would imagine at least some of you might have done ELISA tests. I'm not a lab person. I've never done it. I've watched other people do it. I should be kept away from the lab. I do data analysis. Is price an issue? Not really. It's harder to develop than a nucleic test, but we'll see it in a second. But there's another point of care tests is called a rapid test, and I'll show you in a second, what one version of this is. And here's an example of what you might see, again, as a consumer. This is from Roche. It's a SARS-CoV-2 rapid antigen test. So it's a rapid chromatographic immunoassay intended for the qualitative detection of a specific antigen of SARS-CoV-2 present in the human nasopharynx. So now this tests for your, basically your nasal secretions. It uses different technology called chromatographic immunoassay. And it's intended for the qualitative detection, which means it's a yes or no test. It doesn't tell you how much antibody there is. This test is performed by healthcare professionals using a swab collected from a patient. The results are intended to aid in the early diagnosis of SARS-CoV-2 infection in patients showing clinical symptoms and assist in the initial screening of patients. It has some parameters, specificity and sensitivity that are reported, results ready in only 15 minutes, and so on. Now, here's what one early rapid test looks like. So first thing, it actually, from the consumer perspective, it doesn't look too different from the pregnancy test, which in a way uses the same technology. But it's sort of a point-of-care, cheap, consumer-level way to perform an ELISA-like test. So it was, presented in this paper, and by the way, this is the fastest acceptance. I just noticed it was received 24th February and accepted 26th February. So it was either not reviewed or was so good that it was immediately accepted. 2 days from a receipt to publication. Maybe the reviewers were really fast. So here's what it looks like. It's this lateral flow assay. So you basically, insert the sample and it's pulled across. So you have, presumably you have two types. For this particular test, you can detect IgM and IgG, which is sort of late versus early type of disease. So you have these antibodies and some other stuff that gets put in, sort of flows across, gets attached to labeled conjugates, right? And there are different type of conjugates. So you have the COVID-19 antigen conjugate. So this is something that will actually bind to the antibodies. So this is the tag. This will bind to the antibodies that have specificity for SARS-CoV-2. And there's a rabbit immunoglobulin. This is just a control. So here you have antibodies that capture a human antibody. So this is what happens. You have a SARS-CoV-2 antibody here, among other antibodies. It gets washed across. If it's a SARS-CoV-2 specific antibody, it's going to bind to this complex. If not, it's just going to get washed across, no issue. Now, if it's now, so the ones that are SARS-CoV-2 specific or COVID-19 specific are going to be now with these complexes. So they drift across, they're labeled with something you can see, and then they get captured by these anti-human IgM antibodies, right? And then if you, and they get captured by either IgM or IgG. And this control line, all the human antibodies should be captured here. This is just the line to say, did my test succeed? So you also have rabbit immunoglobulin, which should not be recognized by human receptors. But if you have, if the test worked at all, this line should light up. So this is basically quality control. For example, if you have some flow problems, if there wasn't enough antibody or this step failed, if this line doesn't show up, then the test has failed. This is just basically, did it work as indicated? And it's very simple to understand what the readout is like. So your negative readout. So if there's nothing on the test, it failed, right? If this line doesn't show up. If this is the last line is the only line that shows up, you have a negative test. Then one line plus the control line, you have IgM, IgG, both IgM and IgG. And here's a picture of what they actually look like from the consumer standpoint. So this is what you're going to see as a consumer level test, which is basically a throwaway, immunoprecipitation kind of stuff, which is just packaged in a disposable container and it doesn't require a professional to read it. So here's an indication. Again, this is just mostly for your brief reference. This is another paper that did a comparative analysis of these types of tests. So the reason this is important, because you have a lot of tests on the market. It is important to have them independently validated as far as the quality of detection, as far as whether or not they do as the manufacturer advertised. So you'll have a number of peer-reviewed studies that do just that. So here's an example, for instance. There are 4 tests. So there's the commercial name of the test. This is the antibody that they detect. So the 4 of them are IgG and IgM. This doesn't differentiate. It's just total antibody. Methodology is LFIA, so lateral flow immunoassay. They're all qualitative, so that's a yes or no. Now here they target different. These are different antibody targets. So the first 2 tests look for antibodies that bind to viral nucleocapsid. This one looks to that binds to spike. This just looks for overall IgG. This is what you need: whole blood, either from the vein or finger stick, serum or plasma. They all take the same. This is how much it takes. 10 microliters, so very, very small amounts. They all run in 10 to 15 minutes. They all have reported positive controls. So what rate did they detect? This also has additional information. So for example, it has been confirmed that this test is not going to give you a false positive if you have influenza, adenovirus, respiratory syncytial virus, HBV, HCV, and all the other things. So this is good. You don't want to have a test lighting up as positive if you just have some other viral disease, registration and so on. And, here's their qualitative performance. So let me, walk you through this. These are all antibody tests. So one of the things you want to remember is that they don't work too well early in infection because it takes some time for the antibody to develop. So for instance, if you look at panel A here, there are sort of 3 outcomes that they're reporting. So one panel is per test. So this is old test 2019, nCoV, IgG, IgM rapid test. They've given it samples from patients at different time points in infection. So you have 1 to 7 bin, so week 1, week 2, week 3, week 4, week 5, after week 5. And you can see how well they do, right? So these are all positive controls. So this was 16 positive controls that they fed in. So they confirmed it using RT-PCR and clinical diagnostics. So it's guaranteed that these people have COVID. So all the results should be positive. And as you would expect, early with infection, these tests don't have a great deal of sensitivity. So the first week, for instance, this test is able to detect IgM or IgG. And the first week, it's 20 to 30%. Second week, it increases to 60%. Third week, it's effectively 100%. Fourth and later, 100%. Notice there are different tracks for IgM and IgG. So IgM is thought to be a marker of early disease. So IgM comes up early in infection, and then it sort of transitions out. IgG is a more mature response. So if you see IgM and not IgG, that's probably early infection. If you see IgG but no IgM, it's an infection that's occurred some time ago. But if you look at these tests, basically, the actual dynamics change a little bit. You observe 2 things that are the same. By week 4 or 5 at the most, they detect 100% of all the cases. And then the ones that measure IgG and IgM separately, they have some differential dynamics. IgM decays, so you have some diagnostic power to say if it's an early or a late infection. So if there's no IgM, for instance, with these 2 tests, you can say it's after week 4. And what's shown on the right is basically a reverse survival curve. So this is a way to compare, right? If you look at these tests here on the left, it's really difficult to say which one of them does better than the other. You can kind of eyeball them and say maybe A does better than C because it does better in, A does better than C because in week 3 it detects 96%, whereas in week 3, test C only detects 83 to 87%. But there's actually a specific statistical test that tells you whether or not they are distinguishable from each other. And this would be a form of a Kaplan-Meier curve or Cox proportional hazards model, which you plot here, and it's not the greatest plot. But as you march through time, 0, week 1, week 2, week 3, week 4, and if you're able to track one of these curves, it shows you what fraction of all the cases was detected to this point or before. So for example, this dashed line has 100% detection by week 21, whereas this wide dashed line doesn't reach 100% detection by week 35. And there's a test that can compare them and say there's statistically significant difference between them. Report a p-value, and if it's small, then the tests are actually different. And in this case, for this small sample size, none of these tests do better than the other. The question is, which antibody has the highest binding affinity to COVID-19 antibody? Is it the same affinity throughout relative to concentration? It's difficult to say, right? So I mean, these are just marker antibodies. So you have it doesn't measure the strength of binding at all. It's a qualitative bind, doesn't bind. So you can have differential degrees of binding between antibodies from different individuals, but it's not really important for the test so long as you have some binding. It's not like trying to rip them apart. It's basically binding versus no binding, which also means that you can actually have a fair number of false positives with this test because you can have non-specific binding. Which brings me to this, this is a letter, random letter to the editor from a pediatrics journal, which highlights a very important property of the test, which is you need to, whatever you, as a consumer of the test, you need to be able to interpret what it tells you. And, here's an antibody test that is actually published on the, that's released in the market called the Celix test. It has a sensitivity of 94%, specificity of 96%, which sounds on the surface pretty good. But what it means is that if you're testing for a rare condition, say in your population, only very few people, 1 in 100, has SARS-CoV-2. With these parameters, 80% of the positive results are false. It gets better if you have more and more common SARS-CoV-2, but if you're testing for a rare condition, like 1 to 10%, especially for a very rare condition, the vast majority of positive tests could be false, which is what, the take-home message from this paper is we encourage family physicians to look for appropriately validated antibody tests with an adequate specificity, right? So the ability to find the right infection, even if it means sacrificing some sensitivity. So it might be better to have a negative result that might be, really a positive if you're trading it for the reduction in a very high rate of false positives, right? If 4 out of 5 tests are false positives, it's a useless test. They should encourage laboratories to report test results in a way that reflects the local population prevalence based on widespread testing, which means that the appropriate way to interpret this result is to say, what is my expectation? Do I think SARS-CoV-2 is rare in my population or frequent? Because depending on this background rate, you're going to have very different test performance. If it's say 50% or more, so every other person had SARS-CoV-2, then 4% false positives is perfectly tolerable. But if it's 5% or less, not so much. So this information is needed to help family physicians better inform shared decision-making regarding previous infection and return to work or school. So the one editorial comment that I would like to make here is that next time you see just case numbers or something like that reported without any additional information, it is very difficult to translate that raw number into something that is meaningful, because you can come up with scenarios where, for example, in rare prevalence situations, the number of positive tests could actually be overcounting the real cases by as much as a factor of 5. So you need more information in order to interpret this in a way that is able to inform policy making or even rational decision making from an individual perspective. And unfortunately, I have not seen this responsibly reported. I mean, some people do, like here, for example, you see editorials that appeal to the physicians to do this, but it's up on the physician. It's on the physician to do due diligence. It's not a standard part of the reporting process. So now I'm going to sort of conclude this section by just giving you an early study that was published in, so the paper was submitted in March, it was published in May, which was a detailed study of antibody responses to SARS-CoV-2 in patients with novel coronavirus disease 2019. The importance of studies like this is it establishes a clinical course of the infection. So you have some understanding of when antibodies should be detectable. And, is there a lot of variability between different individuals? And I think this is a good type of paper that I just highlight a couple of things about it that are common to these types of papers. So first, whenever you look at a paper like this, it's important to see how many people were studied, right? So if it's very few people, it's not generalizable. So here it's 173 patients. It's a large study, reasonably large study. So potentially you can learn something good from it. The second thing is, what did they measure? So they measured serial plasma samples. So they took multiple samples from different individuals collected during hospitalization and were tested for a variety of things, for total antibodies, IgM and IgG. The dynamics of antibodies with the disease progress were analyzed. And here's what they found. So among the 173 patients, the seroconversion rates for different antibodies were like so. They make sense. Total antibodies should be greater than either IgM or IgG. The reason for the negative antibody finding in 12 patients might be due to the lack of blood samples at a later stage of illness. So it should really be 100. They just explained why it's not 100. Now, the median seroconversion times was 11 days for total antibody, 12 days for IgM, and 4 days for IgG. The presence of antibodies was less than 40% among people with 1 week of onset, rapidly increased to 100% by the end of the second week. The conclusion is antibody detection offers vital clinical information during the course of the infection. The findings provide strong empirical support for the routine application of serological testing in the diagnosis and management. So this is table 1 is sort of infamous for clinical studies, because if you look at them and if you start noticing, table 1 always tells you all the basic information about the people that were in the study. And that's kind of important because you can see, for example, if it's representative. So it'll break everything down. So there's 137, 173, so many critical, so many non-critical. Was it, gender appropriate? So about 50-50. What was the age representation? This was a Chinese study. So there's additional things like, do we know why they became infected? I'm sure you know that for SARS-CoV-2, the severity of the outcome is significantly confounded by comorbidities, heart disease, obesity, diabetes, things like this. So it tells you what the rate of that is, tells you how many people recovered at the time of the study and were still in the hospital, how many died, some low-level diagnostics like the number of RT-PCR cycles, which you should now be able to understand, and so on. So the question is it okay to couch each sample in the study, even though each patient may have contributed more than one sample? Yes, it's important because you do time series. So for example, here the number of antibodies was an average 3 throughout the course of the infection, because you actually want to understand how antibodies change over time. So you need to take what's called longitudinal measurements. And here's a summary of what all that happened. This is actually, I think it's a great figure. I think it summarizes a lot of information succinctly. So here's panel B, days since onset. So 1, 2, up to 3 weeks. And then the last time point is everything after 3 weeks. And then they have 4 different readouts. So viral RNA, so it's RT-PCR test, and then 3 different antibody measurements. So total antibody, IgM or IgG. And this is how many of the samples tested positive at any particular time point. And you can see this trend that viral RNA, peaks early and then sort of drops off towards the end. Antibodies pretty low in the beginning, and then it peaks and stays high. And then it maybe decays later on. And here's the same information depicted. And here's how many samples were at each time point. So it kind of gives you, so 20 samples were tested for RNA at day 16, for instance. And this is the rate at which things happened. So, the first thing that happened is antibody, then IgM, and then IgG. And finally, the whole point of this is there was a lot of variability across different patients. So this is the type of data that they worked with. For example, here, there's one case, 73-year-old female, non-critical. So they measure 2 things. So they measure signal to noise ratio, which is basically a qualitative readout for antibody. And on the right, they measure RT-PCR cell cycles. So the higher this number is, the less virus RNA there is. So there's what it happens with viral RNA. It pretty much stays on. And then the antibody starts low and gets high. This total antibody stays constant. The IgG just bounces around. So there's not much to be said about this, just to indicate that there are different progressions for different patients. But overall, going back one slide, the summary is like this. So RNA early, decays, then antibody kind of stays low in the beginning, then comes up, and then slightly different dynamics for IgM and IgG. And there's a slide that I mentioned when we talked about RT-PCR testing, and I just bring it back again, sort of put it all together, where before symptoms, you're unlikely to detect things with anything unless maybe you do a sensitive RT-PCR. Then there's early infection RT-PCR is likely positive, and then it decays, and then antibody detection comes on and stays on. And you will have different sorts of sensitivities based on where you sample. So it'll be easiest to find things early in the nose, then virus isolation from respiratory tract because it gets cleared. The bronchoalveolar lavage sort of keeps a lot of it later on, and then stool PCR is when you start excreting the virus. It sort of is the latest. Um, the question is, how was the relative binding signal measured? Would it be like a fluorescent dye? No, it's basically signal to noise. It's just a qualitative readout. So if signal is greater than noise, and depending on your readouts, you probably want it to be more than like 5 signal to noise to call the test. It's not really, it's presented as somewhat qualitative, but I don't think it actually should be read that much. It's basically, detectable or not detectable. It's not the same as viral RNA. I mean, it is correlated with the amount of antibody, but it's not as easily calibrated, say, as RT-PCR. So it just, maybe it's, detectable high, low, but that is not as signal to noise is not so signal to control is not a reliable indicator. All right, so I'm going to now transition to our part 1 discussion of the SARS-CoV-2 COVID-19 vaccine. And we'll start today. Then I'm going to, I assigned you a paper to read that continues this debate or discussion about, what a successful vaccine would entail. And then the next time we'll actually discuss that paper in the beginning. So I'll present my views on it, and then we'll move on to, probably treatment, what's there for therapies at the moment. So one of the things one of the sort of cautionary tales that there has never been a successful coronavirus vaccine. But now obviously is a good time to start. And as you might recall, we had 4 different endemic coronaviruses, the ones that cause the common cold. The first one was discovered in the 1960s or isolated in the 1960s. There was probably never much of an imperative to develop a vaccine for the common cold because it is a disease that very rarely leads to complications, and it just clears on its own. So it's, you feel sick for a couple of days, but then the vast majority of people recover. So it wasn't, a very urgent need. Then, of course, we had SARS-CoV-1 and MERS. There was, especially for SARS-CoV-1, there was a significant effort to develop a vaccine, which we're leveraging now. So it's a good thing it was done then, but there was no successful vaccine, and partly because the virus basically burned itself out. So it was successfully controlled. It didn't spread. The same with MERS. So we don't have a successful coronavirus vaccine, partly because until SARS-CoV-1, we didn't really try. And, we tried after SARS-CoV-1, made some good progress, but then again, there was no incentive to continue developing it. Obviously now there's a massive incentive, so we'll see what we're doing. So vaccination, hopefully everybody knows what it is. It's a process which gives you immunity without the need for an actual infection. You can have slight degrees of protection. So when you think of a vaccine, you probably think about sterilizing immunity. So it's a vaccine that's so successful that immune response basically prevents infection. It doesn't allow the virus to gain a foothold and start replicating at any level. A slightly lesser, but still very useful effect would be protective immunity, which you can sort of think of as the disease might still get there, but it'll be much less severe. So for instance, from the standpoint of an individual human, if I have a vaccine that doesn't stop SARS-CoV-2 from infecting me, but whatever infection I get is asymptomatic, I'm okay with it. It's much better than the alternative, so I'll take that. Sterilizing immunity is the ultimate goal, but protective immunity is useful. Vaccination is the most effective form of dealing with viral and many bacterial infections. There are obviously many successful stories. Perhaps the single most successful story is the polio vaccine. If you haven't looked into this, if your generation or my generation never had to live with polio, but my parents' generation, my grandparents' generation did, and you should talk to people that remember what polio was like. It was a big deal. Measles, very successful. Smallpox, very successful, to the point that you don't get a smallpox vaccine because it's been eradicated. It hasn't been given in the United States since, I want to say, the 1960s. There are also many failures in the vaccine design. So probably the most prominent one is the fact that we still do not have an HIV vaccine, even though it's now been 40 years, close to 40 years working on it. We still don't have a universal flu vaccine, and the ones that we do have is, hit or miss. There's no common cold vaccine. It's a bit of a cheeky question. Maybe we don't need one. But obviously we do desire one for SARS-CoV-2. So when you design a vaccine, there are a number of competing objectives. There's some primary and some secondary, but they're all important in a variety of in combination. So first, the goal of the vaccine is to deliver an immunogen. So you basically have to give the immune system an antigen to target. And you don't want this to be an actual infection. So you want to give it a part of the virus to target, but not the part of the virus that will actually make you sick. Question is, how do we know the smallpox is eradicated? Who makes the decision to no longer distribute the vaccine? Well, technically, the World Health Organization declares things eradicated. So as far as eradicated, there's a definition of what eradicated means. It means, it doesn't mean it doesn't exist in the face of the earth. It's just not detected in the population. And who makes the decision to no longer distribute the vaccine? They're actually regulatory bodies. They're different in different countries. There's one in the United States. I don't remember what it's called, but there is a regulatory body whose job it is to recommend standard vaccination protocols. So there's bureaucracy to deal with this. But as far as eradicated, basically means it's no longer spread. Or there's also I don't know off the top of my head what the actual criterion is, but I'm sure there is a well-defined criteria, say, fewer than so many cases per million population in the last 10 years or something like this. I'm sure we can find it. All right, so it's not enough to deliver immunogens. These immunogens must be sufficiently similar to what a normal infection would cause to stimulate strong and specific immune response. So mostly it'll be B cell, but also T cell. If you want a vaccine to be as useful as possible, you want this immune protection to be durable. So you want to raise it today, and you want it to still be ideally effective until you die. But less ideally, you don't want to have to go back every 2 months to have another shot to just boost your response. You want it to be at least annual. You also want to have a vaccine that avoids side effects and maximizes safety. You don't want to have the cure to be worse than the disease. So these are all health-driven outcomes. But when you're thinking about SARS-CoV-2 particularly, and the scales that are involved, there's also a lot of economics and just production and engineering. So how do you say manufacture 8 billion doses of something, in a scalable and rapid production? How do you deliver it? How do you pay for it? How do you distribute it? How do you store it? There's actually discussions about, we might run out of little glass vials, things that you don't worry about generally, but when you're talking about things that are just scaling up so quickly, there are all these edge cases that are being hit. But this is something that obviously needs to be considered. So there's a story of, there's a one-page story of the original vaccine. So the original vaccine is, accredited to Edward Jenner in the context of smallpox. There's a citation here that if you want to read, it's a 4-page story. It's actually fascinating. I recommend you read it if you're interested, because it sort of gives you, it also puts things in context. So smallpox was a really truly terrible disease that was the scourge of the Enlightenment age civilization. So to quote from this paper, smallpox affected all levels of society. In the 18th century in Europe, 400,000 people died annually of smallpox, and 1/3 of the survivors went blind. So 400,000 people in Europe in the 18th century, scaled for the population, is probably, I don't know, at least 10 times that, probably more, per year, right? And, the 1/3 of the survivors went blind. It was a very deadly disease. The case fatality rate varied from 20 to 60% and left most survivors with disfiguring scars. So I'll make this distinction now and I'll come back to it. So case fatality rate means that you were infected and you developed symptoms. A number of people were infected and cleared it or didn't have clinical symptoms. So that would be infection fatality rate. So case fatality rate means you develop symptoms. Question is, I did see that multiple vaccines trials are in phase 3. Do you think having multiple types of vaccines will help in scaling up vaccine production and global reach. Yeah, I think it will. We'll talk a little bit about it later today. The case fatality rate in infants was even higher, approaching 80% in London and 98% in Berlin during the late 1800s. So this is, 80% infants. It's basically a death sentence. Again, this is something that is shocking to the modern eye. And if you put in the context of, say, SARS-CoV-2, the case fatality rate, depending on how you estimate it, is worst case scenario, maybe 1 or 2%, right? So we're talking about 2 orders of magnitude higher, and in case fatality rate in infants, for SARS-CoV-2 is effectively 0, but here it's 80 to 98%. It's actually an old disease as well, so it was reported in ancient writings, and it was also realized in early on, so about 400 BC, the smallpox survivors were immune to subsequent infection. Which is where they got the idea that maybe somehow you can transfer this immunity to susceptible people. And the way this was done prior to Jenner is inoculation, which stands, the Latin word that's the root of inoculation is grafting. So what you literally would do, you take an infected person, you cut a pustule, with a scalpel or a sharp knife, you'd scrape some out of the pus, and you'd stick it under the skin of an uninfected person. Therefore, you would introduce an immunogen that way. And this was done many times in many locations. This obviously sounds barbaric now because, who's to say you're not going to get the infection in this case? And in fact, people did. So 2 to 3% of those so-called variolated persons died from the procedure or went on to spread the disease. But at the time, it was viewed as an acceptable risk because it lowered the case fatality rate by 10%. So, 2 to 3% versus 20 to 60% is acceptable. Jenner's contribution, were twofold contribution. First, he realized that you don't actually need to give people smallpox. You can give them a milder related disease called cowpox. So here's a picture of the hand from which he actually took. So what he did, he took, so there was this lady named Sarah Nemes in Britain. She was infected with cowpox. So Jenner took a little bit of pus from this pustule from cowpox and grafted it onto the skin of, I think, a 10-year-old boy who went on to develop basically like mild malaise and some discomfort. But then he cleared it, and then Jenner gave him a graft from an actual smallpox disease and discovered that the boy was immune. So those sort of the first sample size of one clinical trial to demonstrate that vaccinations works. And the Latin word for cow is vacca, and cowpox is vaccinia. So Jenner decided to call this new procedure vaccination, in case you ever wanted to know. But yeah, as a historical precedent, I think this is sort of an interesting review because I think it's always important to put things in historical context. Otherwise, we're at the risk of sort of thinking that whatever challenges we live through as a generation, they're unique to us. But yeah, history will argue the opposite in most cases. As far as what vaccines we have today, if you want to look at the complete list, the Food and Drug Administration maintains a list of vaccines licensed for use in the United States. But I took a snapshot from a fairly recent review in 2013, which sort of summarizes different types of vaccines. And let me sort of introduce some different, briefly different terms. We'll discuss them in greater detail in the next couple of slides. But basically, class of a vaccine is the way it delivers the immunogen. So you can have, we'll discuss this again. So I'll give more complete definitions later on. So you can have an activated pathogen, you can have live attenuated pathogen, you can have toxoid, pathogen polysaccharide, recombinant protein, or virus-like particle. Different viruses will be targeted by different vaccines. So these are, for each of the vaccines, there are names of the viruses that are being targeted. Here are examples of market, commercial names of the product. This is when they were licensed. And I think if you've never looked at this, the efficacy of different vaccines actually is dramatically, has a dramatic range. So for instance, rabies, 100% efficacy. MMR, 100% efficacy. But a lot of the things that, polio virus, 99% efficacy. But a lot of the things you will actually see quite low. Tuberculosis, 50%. Influenza vaccine between different seasons might be low 60%. So, 45% for flu block in 2013. So there's a gradient of efficacy. So just because you have an approved vaccine, it doesn't mean it's 100% effective. Typically, it'll be more than 50% effective, but you will see quite a range. Question is, does the license for use in the US mean there are other vaccines not used in the US? It's a great question. So you have, and we're not going to talk about it much. Maybe I'll have an ethics lecture towards the end, depending on how much material we cover, and we'll talk about this. But different countries have very different what's called regulatory environments. So each, and that literally means how drugs get approved for use. So if you take, for instance, what's happening in the United States, Food and Drug Administration is the primary agency responsible for licensing drugs for use and diagnostic tests and, all kinds of other considerations. There's a similar process in the European Union, but if you go to different countries, there's a patchwork of regulations. So for instance, I have no idea, for instance, what the regulatory landscape is like in China or Russia. It's all I can tell you is it's different from what it is in the United States. So you can very easily have a product. For instance, the Russian vaccine is licensed for use in Russia, but it's not licensed, or probably never going to be licensed for use in the United States. The question, there's another great question. Do some of the viruses have low efficacy due to mutagenicity? Yes. So there's influenza sort of as a canonical example of something that's difficult to target with a vaccine because it's a variable pathogen. SARS-CoV-2 doesn't seem to be in that category. All right, so the rest of it is basically going to be a discussion of this paper that literally was published yesterday. It's a great review from an immunologist, I believe at Yale, named Florian Kramer. He's done a lot of work in the area of SARS-CoV-2, both epidemiology and vaccine. He's well respected. So it's a great paper, well written. Highly recommend it. This paper sort of goes through what is the current status of SARS-CoV-2 vaccine development? What does the approval process look like? What are the types of vaccines being developed and tested? How do they differ by immunogen design, delivery, tolerability, efficacy? I mentioned it in the beginning, and I'll mention it here and once more, that depending on which types of immunoglobulins the vaccine stimulates, IgA is basically in your nose, IgG is inside your body. So vaccine only, and which antibodies you raise depends on how you administer the vaccine. So most of the vaccines are done through intramuscular injection. So you basically get a shot in the arm, much like you would with a flu vaccine. Those raise IgG. Vaccines that get sprayed in your nose raise IgA and IgG. So if you're starting with an intramuscular vaccine, it simply does not create IgA response, which means it's not going to be very effective in your nose, right? So it'll probably, what that means, if it's an effective vaccine, it's going to basically give you an asymptomatic infection, but the virus will still get in and sort of have low-level replication and potentially transmission. Production consideration and administration schedule. There is, if you want to go up to date, and see what is the current state of all the vaccine candidates, WHO has a sort of a regularly updated document that's linked here. So at the moment, as of today, there were 38 vaccines in clinical evaluation and 141 in preclinical evaluation. If you go to the document and open this up, here's a partial view. I'll explain some of this. So this is just the top of the list. You will have the name of the vaccine. So University of Oxford, AstraZeneca, for instance, Janssen, Sinovac, Moderna. This is how the immunogen is being delivered. I'll explain that for the rest of the lecture. So non-replicating viral vector, inactivated RNA virus, type of candidate vaccine, how many doses are being trialed, so 1 or 2. What's the spacing between doses? So for instance, here is a shot followed by a booster. 3 weeks later. Here's a shot followed by a booster, what looks like 8 weeks later. Yeah, so Johnson & Johnson got into phase 3 today. Yeah, so there's another study that got into, so route of administration, IM, intramuscular. So all of them goes back to what I just described, that this is IgG stimulating vaccine. And these studies all went through phase 1, phase 1/2, phase 3. So phase 3 is a large scale trial. If there's a link, which means you can go, so NCT is the clinical trial that's being run by the US federal government. If we have time, I'll show you at the very end what that looks like. So there's data on all these phases. And again, I'm just going to, all I did, because this paper just came out yesterday and I thought it was, a really good paper. It's basically just, copy and paste from it. We'll discuss it. And I strongly encourage you to read it, and we'll discuss some more next time. So there's a traditional development of a vaccine, and a lot of this is based, on Western style development. So FDA, EMAs, US and European. But you start with the premise that start to end, large scale production, it takes about 15 years for a traditional vaccine to be developed. Now stop and think about it. 15 years, right? I mean, for most of you, it's not much less than, your entire life. So it's a very slow process typically that goes through a lot of individual phases. And of course, it's a cartoon. So first you have to design and do laboratory tests to see, what immunogen you're going to put in, how you're going to deliver it, how you're going to package it, how you're going to administer it. Then you do animal studies. So you do mice, maybe non-human primates. Then you do toxicology and pharmacokinetics to make sure that the delivery of this vaccine doesn't have side effects. At this point, when all of this passes, you submit a form for the FDA to approve this as an investigational new drug, or an IND, which allows you, which gives you permission to start conducting clinical trials. This is a very slow, deliberately slow and staged process where you go from small scale trials to larger scale trials to very large scale trials. And in order to go from phase 1 to phase 2, you have to complete and show results in phase 1 and phase 2, phase 3. And this is, each phase is a year to 2 years, right? It's a full scale, usually multi-site clinical trial, many patients, many records, statistical analysis, all kinds of things. So, after that is completed, then you present the results of the phase trials, all the phases of clinical trials, to the regulatory agency in what's called the BLA, or Biologic License Application. This takes some time for the FDA to review and approve. I'll say it takes a couple of months. So once you've received approval, if you're a business, now you have to invest time and money and resources in distributing and manufacturing the vaccine, which can take some time. So that's how it usually works. Now, obviously, we're not waiting 15 years for SARS-CoV-2, and there are a lot of places where you can take shortcuts. A lot of it is serendipitous, because this is not a brand new pathogen. We're basically not starting from scratch. So the first clinical trial started in March, right? Very, very early on. So Moderna and NIAID started back then. Some of the Chinese clinical trials started early on. And the reason that you could do this is because you had a lot of the early stage work completed based on the data and analysis from SARS-CoV-1 and MERS. So there's enough similarities between the current coronavirus and the previous coronaviruses, so we can take the technologies and the immunogens and basically just modify them to work with SARS-CoV-2. So that's one stage of compression. Another stage of compression is you can do things in parallel. So you can do phase 1, 2 with overlapping clinical trials, and phase 3 accelerated and immediately on. And the other thing you can do is you take the risk to start production, or at least building production capacity to do things as soon as they're approved. So you're basically stacking things and running them in parallel. Then there's probably, because there's such an impetus to get a vaccine out as soon as possible, the regulatory approval can be shortcut. EUA is an emergency use authorization and goes to regulatory review and release. Unfortunately, And I don't have to tell you this because all you have to do is, read the news or see what's happening. Uh, the process of vaccine design and the motivation behind vaccine design has become heavily politicized to the point where today I was shocked to read that, the governor of New York State, Andrew Cuomo, declared that New York State is going to conduct an independent review of the vaccines because, he doesn't trust the FDA. Uh, so we're creating complications where there shouldn't be any complications. I think, there are enough challenges, but this political layer to the process is unfortunate in my view. There's, like I said, there's enough trouble with the disease and its consequences as it is that we don't need to create more complications and hurdles. The question is, what does it take so long just for approval? Doesn't FDA just have to review the data and efficacy of the trial, or is there other testing that has to be done? There's lots of stages of review. One of the things you need to remember about the medical science is it's deliberately very conservative. You want to move very slowly because the cost of making mistakes is catastrophic. If you miss something, it is, generally speaking, it is much safer to delay the release, to make sure that you review everything and confirm the data, do validation, do subsequent request additional information, do whatever else is necessary, than it is to release a rushed vaccine and then discover that it has a significant underreported side effect, especially if you're going to try to distribute this vaccine to, billions of people right away. So the risk involved here is very significant. So I think the regulatory approval should still occur despite the fact that there's such a public health emergency, because the cost of making mistakes is incredibly high. There's a great cartoon from, I believe it's the FDA, that's addressed to parents, their children's vaccine, that has these nice cartoons, and I like this. It sort of shows you what these phases are like. So phase 1 is, small scale study. So in the order of 100 volunteers. It's designed to address the questions like, is this vaccine safe? Does this vaccine seem to work? Are there any serious side effects? How is the size of the dose related to side effects? All right, so considering that this passes, you move on to phase 2, which is a larger, several hundred volunteers. So basically now you've established basic efficacy and safety. Now you're looking for side effects on a larger sample. If you pass this, you move on to a larger sample to look for more side effects. And here you actually do a proper phase 3 trial where you do efficacy testing, right? So you give some people a vaccine and some people no vaccine and see if the vaccine is actually protective. And you also answer questions, because it's a larger scale study. Is the vaccine safe? Is it effective? And what are the most common side effects? Because one of the things you will see when, once the vaccine is released, you'll have to report on the side effects, what's normal, what's abnormal. The physicians need to know how to react to things like this. And then the FDA licenses the vaccine. Vaccines are made in batches called lots. Manufacturers must test the lots to make sure they're safe, pure, and potent. The lots can only be released once FDA reviews their safety and quality. The FDA further inspects manufacturing facilities regularly to ensure quality and safety. There's also a very, very large and detailed database run by the government called VAERS, or Vaccine Adverse Event Reporting System, which basically collates all the reported side effects from minor to death for all the vaccines. It's publicly accessible. You can search it if you want. You can go. There's no data on SARS-CoV-2 because no vaccines have been licensed in the U.S. But there's sort of an ongoing monitoring system. Sometimes vaccines get recalled if side effects are reported. It's also very rare, but it does occur. All right. So which vaccines are we doing now? Again, going back to the Nature review paper. So here's a cartoon of where the infection occurs, right? So if it targets your upper respiratory tract, the immune response is mostly modulated by immunoglobulin A. Once it gets deeper into your system, it's mostly controlled by immunoglobulin G. So now 3 routes of raising your immune response. Natural infection gets into your upper respiratory tract, travels to the lungs and other tissues, which means it stimulates both kinds of response. So if you're a naturally convalescent patient from COVID-19, you will have upper respiratory protection and lower respiratory protection. Two different routes of vaccine delivery would be intramuscular, where you get a shot in the arm. This only raises IgG responses, which means it protects your lungs, but it doesn't really have much of an immune response in the upper respiratory tract. If you inject the immunogen sort of the natural way, the same way the virus infects into the nose, then you stimulate both types of responses. So ideally, if we want to have a fully protective vaccine, we should look at intranasal vaccination. And there's some in preclinical trials that do this, but all the ones in the clinical trials, I believe now, are intramuscular or intradermal. So this already tells you that whether the first batch of vaccines is not going to be the final word, just based on this consideration. And the next question is how to deliver the immunogen. So vaccine must deliver and present an immunogen to the immune system. Live infection is the most immunogenic in the sense that the immune system can target everything: viral DNA, viral RNA, all the proteins, all the combinations up there above. So it's as many target, sort of the target-rich environment. Live infection is undesirable. I mean, even Jenner realized that live infection with smallpox was not the optimal thing, and that was in the 1700s. So what he did was basically the first, what you would call an attenuated pathogen, where you don't infect with the actual smallpox, but you infect with a much milder virus that raises a very similar immune response. So now there are numerous strategies that have been developed to deliver less than live infection and attempt to stimulate an immune response. There's a panoply of things that you can do. So I'm just going to throw it up here. You can come back and review it, and you can go back to the paper, but there are at least 10 different ways that people are pursuing the vaccine. So what I'm going to do, I'm going to go through and discuss some of them, and then we'll talk about a couple of examples. Sorry, this slide is a duplicate. So at this point, I'm just going to read a couple of passages from the paper because I thought about summarizing them, but they're already succinct, and I don't actually need to reword things that are well said. So the first class of vaccines is inactivated vaccines. So they're produced by growing SARS-CoV-2 in cell culture, usually on vero cells, and then you chemically inactivate the virus. They can be produced relatively easily. This is a standard way to do it. But their yield might be limited by the productivity of the virus in cell culture, and because you're dealing with live virus at some point, you have to do it in very restricted production facilities. So biosafety level 3, BSL-3 facilities. So there are a couple of vaccines in China that have been developed using inactivated vaccines, in India, and the Research Institute for Biological Safety Problems in Kazakhstan, the former Soviet Union. These vaccines are usually administered intramuscular and might be adjuvanted with alum or other adjuvants. I'll explain on the next slide what that is. Benefits of this vaccine is that the whole virus is presented to the immune system, which means the immune responses are likely to target not only the spike protein, but also the matrix envelope and nuclear protein. And there are several inactivated vaccines that have entered clinical trials. What can be some of the reasons why some vaccines can be ineffective? Let's go through the vaccines that they're trialing now, and we'll come back to this and try to answer it. So vaccine adjuvants is basically a broad term. You can think of them as chemical compounds that make the vaccine more effective. I'll give you examples on the next slide what they are. But the fundamental idea is that you always have this balancing act. So the strongest immune response you can get is by having live attenuated pathogen, right? So this is the gold standard. You basically take a virus, make it weaker, and infect the person with it. There are dangers with it, right? Because it can run away, it can actually cause a significant infection. So people are not really doing live attenuated pathogens that much. They're sort of doing either killed viruses or parts of viruses. The trade-off here is that they're not as immunogenic. So the immune system might clear them a different way without raising a response. So what you do, you add adjuvants which stimulate the immune response in a certain way. And they were accidentally discovered in the late 1920s. There's this great chart from a paper that's cited here. It says, so this is the history of the vaccine. So smallpox, if you remember, as we discussed, was the late 1700s, and you have rabies, typhoid, cholera, and plague. There was no adjuvants back then because there weren't, nobody knew that they existed. Then adjuvants were discovered in the late 1920s, and then basically about half of the vaccines from that point on are adjuvanted. So some of the examples And the first one was discovered was actually aluminum. Aluminum is salts mixed with antigen, and it has this, the effects is that it actually increases local inflammation, improves antigen uptake by antigen presenting cells, and acts to increase antibody production. So all it really does, it kicks up your immune system into an activated state. So it simulates the presence of inflammation, which then triggers the entire cascade of immune responses. There are other less pedestrian ones, but aluminum is to this day used as an adjuvant. And that's, if you ever watch any of the anti-vax propaganda, a lot of it will have to do with the presence of adjuvants as, non-naturally occurring mechanisms and, substances. There's actually a good reason for putting them in. So the next type of vaccines would be live attenuated vaccines. So these are sort of the closest to the live infection you can get. So you will, sort of euphemistically speaking, generate a genetically weakened version of the virus to replicate to a limited extent, cause no disease, but induce immune responses that are similar to the immune response induced by the natural infection. There are a variety of strategies about how this is done. Obviously, you want to make sure that the virus doesn't become as infectious as the full-on SARS-CoV-2. So there are different techniques that you can employ. For instance, you can passage viruses to adapt to unfavorable conditions. So before you give them to humans, for instance, you passage them in, pig cells or mouth cells, or you grow them at lower temperature, so they become adapted to growing in environments that are not like the human body. You can also rationally modify them. So you can, for instance, delete some of the genes that are, some of the ORFs for SARS-CoV-2. Or codon deoptimization is a technique where you basically keep the amino acids the same, but you change the nucleotides in a way that makes the virus less efficient at replication. So for instance, SARS-CoV-2 is very CG poor. So it has most of the nucleotides in its genome are Ts. But you can, for example, replace them with Cs and Gs, maintaining the same amino acid, and it's going to make it less efficient. An important advantage of these vaccines is they be given intranasally and induce mucosal immune response, which is, again, desirable. In addition, since the vaccine virus is replicating in the vaccinee, the immune response will likely target the entire gamut. There are also disadvantages. The primary one is safety concern, and the need to modify the virus, which is time-consuming and technically challenging. There are only 3 live attenuated vaccines that are currently in preclinical development, including one that is attenuated by codon deoptimization. So the other vaccines are now, so these are the 2 types of vaccines that basically try to inject the entire viral genome, so give your immune system as many targets as possible. The next set of vaccines is sort of more modern, and it basically takes one part of the virus and just presents that part of the virus. So recombinant protein vaccines are one such class. It's actually a very popular class. They're easy to manufacture, for instance. So recombinant protein vaccines can be divided into recombinant spike vaccines. So they take the entire spike protein, recombinant receptor binding domain vaccines, so just a part of spike, which I'm sure you're now deeply familiar with because you read the article from last time, and virus-like particle vaccines. These recombinant proteins can be expressed in different expression systems, including insect cells, mammalian cells, yeast, and plants. They can also be expressed in E. coli. Depending on the expression system, you can vary the yield. You can also have some issues like post-translational modifications. You can also modify the protein, like delete the cleavage side, add some mutations to make the protein more stable, more immunogenic, inclusion of trimerization domains, and so on. So these are just technical quirks. The advantages of these vaccines, they can be produced without handling the live virus. Some recombinant vaccines, like Flublok for influenza, have been licensed, and there's considerable experience in producing them. There are disadvantages, however. You're effectively expressing an artificial version of the protein, so it might be hard to express, which means you will have low yields and might have inefficient production. If you don't express the entire protein, only express the RBD, you effectively target your immune system to only one part of the virus. If the virus develops mutants in RBD, the vaccine is ineffective. There are many recombinant protein vaccines in the trial. Now you have a different class of vaccines, which are called replication active vectors. They all do the same thing. They take a different virus that is not SARS-CoV-2, that is not particularly dangerous at all. They've been tried in many other conditions. And they engineer one part of the SARS-CoV-2 into that vector. So it literally is a vector in this case is something that transmits that protein, delivers that protein to the immune system. They're typically based on another virus that has been engineered to express the S and has been disabled from replication in vivo by deletion parts of its genome. So it means it can infect the cell, but it's not able to replicate. So it's one shot off, right? It gets into the cell, releases payload, it's done. The majority of these approaches are based on adenovirus, but there are different types of viruses like Ankara, Vaccinia Ankara, human parainfluenza, adeno-associated virus, Sendai virus are also used. They are delivered intramuscularly, enter the vaccinee cells, and express the spike protein to which the whole system, immune system responds. The advantages are you don't have to handle live virus. There's significant experience with producing larger quantities of these vectors, and they stimulate both B cells and T cell responses. A disadvantage is some of these viruses are impacted and partially neutralized by pre-existing vector immunity. So your immune system can actually kill the parts of the adenovirus that it recognizes and never raise the response to the desired protein. Vector immunity can be problematic. The pride boost regimens are used. So basically there's some complications in terms of making sure that the right immune response is raised. So the Oxford vaccine is one of those examples. Janssen is also phase 3 study, not recruiting yet. They're recruiting now. So as of today. So these are replication active vectors. Slight modifications, you can have replication active vectors. It's the same thing. Attenuated vaccine strains of virus have been engineered to express a transgene. In some cases, animal viruses that are not replicated efficiently cause no disease in humans are used as well. Obviously, here you have some cycles of replication, so you have more opportunity to raise the immunogen. You can have inactivated virus vectors, similar thing, just the virus is completely inactive. And now there's a completely, so these things have never been tried before, the new two classes, DNA vaccines and RNA vaccines. So everything before is either based on the viral particle and the proteins. This basically is designed to deliver DNA or RNA into the cell and then make the cell synthesize the protein. So DNA vaccines are based on plasmids, which are used by bacteria to basically exchange gene cassettes. So they're very useful for transportation of small genetic payloads. Because of that, they can produce in massive scale as bacteria. Typically, these plasmids will contain mammalian expression promoters. So you'll basically just add the S gene payload, stick it into the cell, and make it generate the S protein. The huge advantage of these technologies is you can scale them up dramatically in E. coli. It's a very stable molecule, plasmid DNA, easy to store, easy to transport. However, they have issues. Once they've never been tried before successfully, and they also show low immunogenicity and have to be delivered via delivery devices to make them efficient. So you can't just take them by injection. So you need to have electroporators for their use. There are currently 4 DNA vaccines in clinical trials. And RNA vaccines, which is what Moderna is doing, this is also brand new. The genetic information with the antigen is delivered instead of the antigen itself. The antigen isn't expressed in the vaccinee cells. You can either do mRNA with modifications of self-replicating RNA. mRNA usually requires high doses of self-replicating RNA, which amplifies itself. So mRNA, deliver once, make the protein. There's no replication cycle. The RNA is usually delivered in these cool lipid nanoparticles, so just little fire-like vectors. So these are brand new. We'll see what they do. They could be very cool, but they've never been tried before, and there are potentially complications that we don't appreciate. So there's a lot to be learned about them. But if this platform shows success, then opens up a whole new avenue for really rapidly developing new techniques. Because if all you have to do is design the mRNA to deliver, then you can adjust it to express whatever you want very quickly. You don't have to inactivate anything. You don't have to insert things into vectors. You don't have to grow inactivated viruses. So this is potentially a modern approach to vaccine design. And finally, I'll just briefly mention, so there's this plethora of different vaccines. They're going through different stages of trials. And just as an example, phase 1, to evaluate a vaccine, you do animal studies. You do it in mice and then in non-human primates. If these tests succeed, then you move on to clinical trials. So, ask the questions like, is the vaccine immunogenic? Does it generate antibodies? Does it lead to seroconversion? And how good is the response? So if it does something, then you ask, is it well tolerated? Do people have side effects? Then if all of this is not actually good at stopping infection, it could raise a response, but this response might not be protective. And then is this response durable? So, if you read this article, there'll sort of be these types of passages there. So the Oxford AstraZeneca and Serum Institute of India vaccine is based on a replication chimpanzee adenovirus. So here's an example of a non-human primate study. This vaccine was tested in rhesus macaques in a prime only and a prime boost regimen. So prime only means you give one shot. Prime boost means you give one shot, and then wait a little bit, and then boost it with a subsequent shot. You give people a lot. You give these monkeys a lot of viral particles. So 2 times 5 to the 10 to the 10. That's 25 billion viral particles given intramuscularly. You give prime and boost in 4-week intervals. And then you wait 4 weeks and challenge them with an infection. One of the things you will see is this typical readout, neutralizing antibody titers, 1 to 5, 1 to 40. These are dilutions. So 1 to 1 means you take undiluted serum. This is the maximum concentration. 1 to 5 means you dilute it by 5 and it's still effective. 40 means you dilute it by a factor of 40. So the more you dilute it, the more potent the antibodies are. So 1 in 5 to 1 in 40 is pretty weak, maybe moderate, and they're increased with boost. But both groups were protected from lung disease post-challenge, which means it was effective, and also mostly from viral replication in the lungs. And here's an example of how you would look at an actual clinical trial result. So this is the same clinical trial, the same vaccine candidate, but now it's in clinical trials in people. So based on their long-standing experience, the University of Oxford, together with AstraZeneca, blah, blah, the recently reported preliminary results from a phase 1/2 single blind. So single blind means the patients don't know what they get. So, placebo or vaccine, but the doctors do. Randomized controlled trial in 1,077 participants aged 18 to 55. You can go look up this trial. The vast majority of the participants in the vaccine group received a single dose of 50,000 viral particles. But a small cohort of 10 individuals who received a booster dose. They controlled it by comparing with the meningitis vaccine that used the same delivery mechanism, used the same vector to basically compare side effects. Antibody responses were tracked using several binding assays, blah, blah, blah. To determine neutralizing antibody responses, a subgroup of 35 individuals was analyzed. And here what they have. So they have a 25 post-day vaccination titers 1 and 218, which is pretty good. 100% seropositivity, 151, depending on the measurement, 91% positivity, so on. A booster dose increased the titers in ladder 2 assay significantly. Interestingly enough, they noted pre-existing immunity to SARS-CoV-2 in 4% of the individuals. They also measured cellular immunity, and they report some side effects like fatigue, greater than 70%, headache, feeling feverish or having an elevated temperature was relatively common. The booster dose seemed to be well tolerated, but since it was only given to 10 individuals, this has to be taken with a grain of salt. Overall, it had a worse safety profile than licensed meningitis vaccine used in placebo arm independently without, fever reducer, and so on. And here's a summary table of the available overview. So just to know how to read it, here's a company that designed the vaccine. There's the vaccine that was being tried. Here's the dose range and how it was given. So 3 to 6 micrograms intramuscularly, 2 times, 2-week booster, 4-week booster, viral particles intramuscularly, micrograms. Qualitative neutralization after prime. So this is first shot, if it's a boosted, and then neutralizing titer after booster. So this is, it's all increased. Whether or not T cell response was measured or not. and whether or not a clinical trial was registered. All right, so we'll resume next time because there's a lot still to cover. But there's this great review article that was published recently in the Journal of Vaccine, which sort of raises some discussion topics about how we should be thinking about vaccine design in the context of SARS-CoV-2, especially when it comes to the balance of B cells and T cell. Two more questions. Could self-replicating mRNA potentially lead to prion-like aggregation? Don't know. Prions are typically misfolded proteins, so there could be, mRNA could potentially do all kinds of things that we don't fully understand, because RNA is a complex molecule with a lot of function. So I would like to, that there are potentially issues that we're not aware of, but I would like to think that this would all be quantified in trials. Are the lipid vectors you mentioned before basically liposomes? We'll talk about it. I'll start describing it again next time. I don't want to keep you guys past 8 o'clock. So this paper is great. Highly recommend it. Is, like I said, a lot of discussion points that we'll come back and talk about them at the beginning of the next lecture. Thank you very much, and I'll see you in a week.